and evolution of geological cycles as well. As you can see, the Earth is not a static planet. That's the specialty, actually. It's maybe more important than distance to sun or the size when you think about it. The fact that Earth is a very dynamic place, continents are being formed, being lost. That's quite important. Very good question, Efe. Yeah. How similar mineral compositions of Earth and Mars? Very good question. I don't know the exact answer, but as far as I think that's one of the objectives of the current mission that they have sent to Mars. It's going to collect samples and bring back those samples so they will understand more on the mineralogy. But at least from, I think, surface orbiters, they estimate that a limited set of minerals exist on Mars. But of course, they don't know what's in deep rocks of Mars. So at least the current forgotten name of the rover that they have just landed on Mars, but it's going to drill, the rocks, collect samples. It's actually in a kind of a lake basin, as far as I'm not wrong, and it will work in a stratigraphic section, so sampling different periods in Mars. So I think they will find our knowledge of Mars will amplify a lot. But how similar, that I can't tell. But my first guess would be Earth is a lot more diverse in terms of its mineralogy. But the basic minerals are the same, if you're asking about that. I mean, iron oxides, I know that iron minerals are the same, at least the oxidized form of iron minerals. They are similar to what we find on Earth. But we don't know, for example, carbonate minerals. We make if there was some sort of life, there must be some sort of carbonate precipitation. I think that's one of the chief ideas of this mission, which is out there at the moment. I think they will also do some in situ analysis, so they should be transmitting data in a year or so on the first analysis of those drills. But good question. I know that in other solar system bodies, they find also different minerals, silicates, different iron minerals, silica minerals. So in a way, the core set of minerals probably is the same, but the Earth happened to have a lot more species of minerals, of course, partially because of life. So any other question? Thank you, Efe. That's a great question, really. I didn't think about comparing them. Another way to look at evolution, now a little bit coming back to maybe to relate this to other courses as well. A major event with respect to evolution, of course, well, there is the question of origin of life, which is not very well constrained, but maybe for you more relevant is the evolution of eukaryotic branch, which happened actually, as you can see here, in this era called Proterozoic. Okay, so if you recall this Proterozoic oceans, I have another type of graph showing you that. That's from a more recent paper, which I like more because you can see the chemistry at the same time. That's the ocean chemistry. That's the atmospheric chemistry with oxygen levels. So the idea here, you see the major life events actually just shown. The origin of life probably happened around here. We don't know how, but these are some major evolution innovations. Nitrogen fixation, for example, is, you know, life wouldn't be able to diversify this much if nitrogen fixation weren't invented. That happened quite early, 3.8 or 3.3 billion years ago. The origin of oxygenic photosynthesis is also dated a little bit earlier than 3 billion years ago. Well, there was probably unoxygenic ways of doing photosynthesis, but they were not, those mechanisms were not producing oxygen, but maybe they were producing sulfur. I'll come to that in a moment in discussion of chemosynthesis, but it's possible to not produce oxygen and do photosynthesis, as is still the case in the Black Sea or in some lakes. Anyhow, as a result of especially nitrogen fixation, this was a major innovation, maybe supported the diversification of cyanobacterial lineages, and maybe it enabled really this high amount production of oxygen, which then injected enough oxygen to the atmosphere to fully oxygenate it. And that happened, as you know by now, around 2.5 billion years ago. And the surface of the ocean then became oxygenated with nitrate as a stable nitrogen form. So you start to have some oxygen-nitrogen cycles being set up. The idea was that once the atmosphere was oxygenated, this oxygen attacked to the sulfur bound on rocks on continents. Okay, so oxygen, once it's produced, it's a very reactive substance actually. So it can go and reduce or oxidize some of the rocks containing sulfur. So a lot of sulfur then was mobilized from continents to oceans. And this sulfur, since most of the ocean was anoxic still, it actually contained, or it led to the evolution of so-called sulfur-reducing microbes as well. And then as a result of their activity, hydrogen sulfide started to accumulate. So it's interesting, the presence of hydrogen sulfide indirectly is also linked to the oxygenation of the atmosphere, evolution of cyanobacteria, which is then probably very much linked to the innovation of nitrogen fixation very early on, which is in a function of availability of iron and molybdenum. There's a whole lot of going on in that front, but you see all these things can be very much interlinked. But what's funny is that between, then you may ask why the oceans did not oxygenate, here fully, but only in the past 500-600 million years. But that's if you find the answer, you should publish it in Nature or Science, to be honest. It's probably a combination of life, geochemistry, geology, but for some reasons, all of those factors kept the Earth system quite stable for about even 2 billion years, 1.5 billion years. There was oxygen in the very surface of Earth atmosphere, and of course the oceans. Then if you are close to the margins, you had hydrogen sulfide beneath this oxygenated layer. If you are far from margins, people think that it was more suboxic, so-called suboxic, did not contain much sulfur, but still it was anoxic. So the whole global ocean was like this. It was like, in a way, present-day Black Sea and some other low oxygen systems we find in different parts of the global ocean. But what's interesting is that, so until then, maybe archaea, the two major branches of life, two of the three major branches are archaea and bacteria. Probably they were evolved, they were around, but eukaryotes were not yet around. And people think that maybe you know about this endosymbiotic theory, but somehow it was a single event, the origin of eukaryotes, single event, a endosymbiosis of a bacteria and archaea. As you know, one became mitochondria, etc. But anyhow, the origin of eukaryotic organisms are now dated to around 1.5 to 1.2 billion years ago. So the origin of eukaryotes happened, in a way, in the oceans when the ocean was like present-day Black Sea. So that's quite interesting. And it's a very open question, why that happened in that particular time. Is it more like contingency of different things, or we don't know, but people can now reliably date these things through also molecular methodology. I think it's a combination of evidence from geochemical isotopes, but also now there's a lot of new work on molecular genetic side, really molecular phylogeny work, really, you know, using the molecular methods as a clock in a way to go back in time. I'm sure you will have some wonderful lectures about that too this week. If so, let me know. I will also be sitting in those lectures. But that's pretty much it. And after, in the past, then 1 billion years or so, then we had the 3 major branches of life. Then once the eukaryotes were also around, somehow the diversification amplified. Even the multicellular origin of multicellular organisms, it's probably can be dated to this very late stages of the Proterozoic oceans, when the ocean was still largely anoxic. Some even argue that the first multicellular organisms, they evolved in an anoxic environment. Anyway, there are new papers about that. And then past 500 million years, then the ocean was pretty much like today. But for example, diatoms, I'm sure many of you know that they are quite recent species, for example. Now they dominate with coccoliths many part of the ocean and lakes as well. But evolutionarily, they are relatively young, looking at this scale. Of course, if you're working on other organisms, maybe diatoms can be still very old, but in this scheme of things, they are recent evolutionary actors. Okay, so you can see, I mean, all these, what is important is that all these mechanisms, metabolic, let's say, pathways, nitrogen fixation, oxygenic photosynthesis, nitrification, denitrification, this, that, they were actually well in place until we started to have some eukaryotes, eukaryotic evolution possible. So all these basic metabolic functions evolved actually much earlier. Even in the Archean, most mechanisms evolved in the Archean. And with oxygen, of course, new mechanisms evolved. But then that's it. I mean, at the moment, probably we are not evolving any more new mechanisms, but we are more, evolution is spending its whatever, how do you call it, potential. It's more on inventing new organisms, maybe higher level organisms. So that's another open question. But all these data now come from molecular biology, actually, looking at gene histories of different lineages. All right, so with this, let's come to a little bit, as promised, maybe I'll keep this a little bit short and move to the extreme deep environments, which may be interesting for you. Biogeochemistry, as I said, I would say it's more about modern understanding of modern systems, modern Earth systems, elemental fluxes. Here you see, for example, a basic model of oceans. We can consider oceans as a surface ocean, mid-waters, and deep ocean. Deep ocean is mostly and typically defined as environments deeper than 200 meters. 200 meters is like the maximum depth that light can penetrate to. But of course, that's the ideal conditions in very clear particle-free waters, which is not mostly the case. In a typical coastal marine environment, I mean, we observe light penetration depths maybe as low as 20 meters, 30 meters. It can even be much less if you are really in a river mouth, for example. But in a typical open Mediterranean Sea, you can have more than 100 meters very easily. So deep sea is quite, it's dark. It's primarily defined by its darkness and high pressures. Okay, a lot of course, commonality between seas and lakes, the way any water body actually, with regard to its biogeochemistry. This definition is from a deep lake, for example. So those of you in knowledge are quite familiar with the photic zone, to which light penetrates. But there's also, depending on season, of course, you may have an anaerobic zone or aphotic zone. And then, of course, any water body, oceans or lakes, they will have some sediments beneath. Most sediments are anaerobic. That's almost everywhere that's the case, because there's so much going on to consume oxygen there that most sediments are anaerobic. And we are also working on microbiology of the anaerobic sediments. So Matt Jan, who is listening to us today, he's also developing expertise on that. So this sort of idea of losing oxygen, then triggering anoxygenic or anaerobic processes is called, it's called a redox gradient. Redox meaning reduction oxidation in chemistry, but basically you change the chemistry. I mean, you switch from oxygenic processes to anaerobic and oxygen-free processes. And then a lot of interesting stuff happens. The chemistry changes. Communities, microbial communities, totally changes after that. So that's why we call these sort of gradients redox gradients. You don't need to have a water body to have redox gradients. You can have a particle, for example, organic particle just hanging around in water. It can sit in an oxygenated water, but people have found using microsensors that even these sort of particles, they have a redox gradient towards its center. So it's a radial gradient in this case. So in an apparently oxygenated environment, because we had those cases where we have sampled particles, looked at metagenomics of those particles, and we have found a lot of genes about anaerobic processes. That's from those particles. The interior of these particles probably inhabited by anaerobic microbes. Even in an apparently very, very oxygenated environment, you will have anaerobic niches, basically. That's gonna increase microbial diversity. Of course, this happens in hot springs, as we will see in an hour, deep sea vents, or even hypersaline lakes. There are even lakes, hypersaline lakes in the bottom of the sea, by the way. The sea is already salty, you know, but it happens if you have some salt rock dissolution. That happens in Mediterranean. You can have very, very salt water, brine, it's called brine in those cases, but hypersaline water is just sitting as a lake on the seafloor. That's fascinating. And those lakes also harbor unique communities around them. Just I saw in Twitter last night that offshore Israel, they just discovered a huge hypersaline seawater lake, seafloor lake with a lot of chemosynthetic tube worms, etc., which we probably have a lot of these things in the Eastern Mediterranean. Anyway, so this is an overview of different modern environments for you. That's just a sketch of a lake sediment again. If it's iron rich, you will have different zonation. So even the anaerobic zones, they will have subzones in between. You may be asking, okay, what drives this sort of zonation? The idea is, let's first actually look at this. The idea is that you can, of course, deplete oxygen, but there are other ways of, let's say, doing respiration. And you don't need to always use oxygen to do respiration. You can use other ways of, actually, let me do this first. You can use other let's say, oxidants for respiration. So respiration in a nutshell is about eating food, making organic carbon to CO2. And why we do that, essentially the idea is to shuttle electrons in your mitochondrial membrane, so that those electrons hop from one protein to another with the eventual idea of generating ATP, right? And once you are finished with those electrons, you need to dump them somewhere. You cannot just store them in the cell. You need to dump them somewhere. Oxygen is a great dumping ground. It's the best dumping ground for electrons. So imagine oxygen as like that. So that's why we breathe oxygen. We dump the electrons that we get from the food. So we dump these electrons to oxygen. The oxygen is then released as water. Then is breathed out as water. And the whole process generates ATP, which is energy. So that's oxygenic respiration. You can also respire sulfate as the microbes in Black Sea do. You can respire sulfate. The same thing, you eat organic carbon, you send your electrons to sulfate. So that's all, there's oxygen in sulfate. Then this is reduced to become more electron rich. This is S minus 2, it's hydrogen sulfide. Then this can lead to a lot of, mineral formation as well. Sphalerite is one of them. You can also breathe iron. Iron has 2 oxidation states, iron 3 and iron 2. You can breathe iron 3 and dump your electrons to iron 3, which then becomes iron 2. Then it can be deposited as different minerals, or it can hang around in solution. So there are different ways. Life evolved all kinds of ways of, even there are microbes breathing uranium in contaminated sites. So different oxidation reduction couples, they can be used for breathing. So that's respiration. Similar reactions, these sort of reactions are called redox reactions, in which an electron exchange is of interest. You can also use the reverse of these reactions in a way to do synthesis. Okay, so if you are doing synthesis with using light, then that's called photosynthesis. You can use chemical energy, that's called chemosynthesis. So this simple table is just very simple, but it brings together many different metabolic ways, lifestyles. So you can, again, when during your readings, during your own thesis work, you may end up reading photolithotrophs or the chemolithotrophs. So what are they? It's coming from this. So it's a shorthand definition of what those communities are doing. Where are they getting their energy from? From light or chemical compounds. So photo or chemo. Where is your electron coming from? You need, as I said, the basic way of cell operation, you need to make ATP, and the way all branches of life, a shared trait is, in a way, the way we generate ATP through this cell, setting up gradient, ion gradients through an action of a series of proteins. And then these ion gradients, it drives actual ATP formation in specific enzymes. But you need a flow of electrons to operate that system. Every organism is a shared trait, which is probably giving a hint about the first form of life. But electrons should be coming from somewhere. In our case, we are organotrophs. We get our electrons from food. But as you will see in a moment, there are also lithotrophs. They get their electrons from inorganic compounds. And of course, the idea is also to assimilate carbon, right? We need to build biomass to do what we do. And if you are obtaining your carbon from CO2, as plants do, or as cyanobacteria do, then you're an autotroph. But if you're like us, if you obtain your carbon from food, then that's heterotrophy. So we have all kinds of combinations currently on Earth. So, and as I said, this summarizes up for your respiration. But another, the reverse of respiration is synthesis in a way. And for synthesis, maybe you are more familiar with photosynthesis, but as I said, you can also obtain your electrons from lithogenic sources, from inorganic sources, and then you don't need light to convert those sources to energy. You can also power your reactions with chemical energy, and you see different microbes have invented ways of doing this through chemosynthesis. So now, in this case, this is called chemosynthesis. There are different ways of doing chemosynthesis. For example, sulfide oxidation by nitrate, special bacteria doing that, methane oxidation by nitrate, anamox, nitrate can be oxidized by nitrite, methane oxidation by sulfate, etc. So all kinds of chemosynthetic pathways exist. As long as there's a suitable chemical reaction giving suitable energy, theoretically it's possible. So what determines this sort of then let's say chemosynthetic possibilities or respiratory possibilities? It's thermodynamics, just to make you familiar with a little bit about this concept, the concept of free energy, which can be calculated for any life, any metabolic reaction. If it's negative, it's good news, then your reaction can proceed, okay? But thermodynamics, in a way, tells you what is possible, but doesn't tell you how fast. It tells you that a certain reaction can be metabolically energy yielding, but over how many years? Over 1 billion years, or over a fraction of a second? That's the science of kinetics, actually, which studies these sort of things. For example, one reason we have enzymes is that we want to catalyze, we want to speed up certain energetic reactions. So that's why we have enzymes. And enzymes basically, they actually speed up things. They don't maybe change the overall free energy yield, but they shorten the time to reach to the activation level to achieve that reaction. So enzymes do this. That's why it's not only when we study biogeochemistry, we of course look at distributions, but we also need to have an idea about how fast things can occur to be able to propose it as a metabolic reactive pathway. Diffusion times. What is interesting is that I've talked about diffusion before. And if you're, of course, now we are more speaking about microorganisms, bacteria and archaea, maybe unicellular eukaryotes. They are not that mobile. Most of them are not mobile at all. They are just in whatever environment they are, they move with that environment, or they don't move at all. So diffusion of resource in this case, oxygen, glucose, whatever, nitrate. So diffusion of these resources to this example is actually can be very it's the life and death for this bacteria. Here you see actually how slow can this be, how slow can this be in certain environments. For example, if you are away, if you're a bacterium, if you are 1 millimeter away from oxygen molecule, it takes about 5-6 minutes for all these oxygen molecules to diffuse to you. So that's, if it's 10 centimeters, 40 days, so quite slow. So you need to be locating yourself in very similar environments. But a recent body of work, again, emerging work, for those of you interested in these sort of things, cable bacteria, for example, they have evolved, some bacteria evolved to of course, it's well known that bacteria, microorganisms, they form filaments, you know, they can form long stretches of networks, basically cooperating. But in this case, it's very interesting because they discovered that bacteria formed cables, you know, truly cables, conducting cables, vertically connecting the oxic zone of sediments with the lower anoxic sulfidic zone. And what is funny is that in the cells making up of these filaments, only in a way the bottom and top partners, they were making some use of, you know, they were making energy, but the ones in the middle, they were just conducting electrons. So people are still trying to figure out why the hell they are doing this. So they're apparently doing this for the good of the whole filament, but probably they have some intricate ways of sharing energy, the generated ATP, we don't know yet. They're looking at, there are several ERC projects looking at this. But this is just happening in coastal sediments. I mean, probably this is a very widespread phenomenon. So even in the microbial world, they have figured out ways of overcoming diffusion barriers, in this case with cable bacteria. So yeah, I have more about oxygen. I see there are a couple of questions. Okay, maybe first I'll look at, we can read Shewal's question. So the time scale, oxygen became full, even deep sites. Let's go back to that. I think you are speaking about this. Yes, before that, it seems there are durations that oxygen integrated deep oceans. Yes, very true. Good catch. How can it be that possible? What are the reasons? Yes, first of all, it's quite possible. Uh, Ocean is a dynamic system. I mean, you can have certain Earth system conditions which may temporarily increase oxygen production, right? For example, that's what happened. That's more recent, but I think it was, I think, in the Devonian or at one point, the atmospheric oxygen levels even reached 30%, which led to evolution of giant spiders, right? Different. I think we have an entomologist among us. Maybe you can comment as well. But these things are not static, first of all. So it's quite possible to fully oxygenate the whole ocean, and then a few 100,000 years later, you may lose your oxygen. That's what's happening now. Actually, the oceans are losing their oxygen because of global warming and anthropogenic influences. So first of all, there's a very dynamic situation associated with our oceans. But what happened in those intervals, I think you were asking about the beginning of the Cambrian, this figure just shows you that actually the full oxygenation didn't happen in one day. Okay, it's just a visual identification of maybe there were full oxygenations, but then it was not stable. For example, people argue that hydrothermalism increased, especially after 0.8 billion years ago. There's evidence that a lot more hydrothermalism occurred with more iron to coming and consuming oxygen. So that may be one reason. It may be biology as well. If you have a lot of too much biology in your surface, then they will sink and consume your oxygen. So probably a combination of those led to return of anoxia to the oceans from time to time. But after, in the past 400 or so million years, it has been rather stable. But now we are now in an era of losing the oxygen from the oceans. As I was just coming to that point, actually. Next few slides. How can you identify hypersaline lakes? That's a combination of bathymetric studies, seismic and studies. You need to collect data from seafloor, and then eventually you need to dive there or send an ROV and observe the hypersaline lakes. You look at basically different remote sensing. It's in a way remote sensing, but it's not with satellites. You do remote sensing of the seafloor and the subsurface beyond it. Probably different salinities, they have different reflection of different signals. Okay, Melike is asking, Kanal Istanbul affect Marmara Sea in terms of sulfuric water coming from Black Sea. The Kanal Istanbul, it's very shallow. Actually, we don't know anything about this canal, for example, how deep, but its depth is probably not going to be that deep. The sulfuric waters in the Black Sea start around 120 meters or something like that. So, and the sulfuric waters are further offshore. So, in the point where they have this canal, it's also oxygenated still continental shelf Black Sea water. So it would be oxygenated part of the Black Sea flowing into the Marmara. And the real effect of Kanal Istanbul will not be that at all. I mean, that's what you indicate is a good point, but there are a whole lot of other issues, especially for Marmara Sea. But essentially, it's quite possible. The oxygen in that was quite actually my next course, which I don't want to also take too much of the time with this to make space for deep sea ecosystems, but I was just going to make this point that changes in geological past, of course we don't understand how, but we can look at current global change to understand that. And with respect to oxygen, you need to again know that as ecologists, that it's very easy to lose oxygen. First of all, in the ocean, we don't have much oxygen to start with. It's not like atmosphere. In the atmosphere, it's 21%. In the ocean, it's much, much, much less. Because why? It's a gas. It's chemically, the chemical reasons. It's a gas, tends to be in the air, and a very small fraction of this can be dissolved. And you know, gases tend to be more dissolved in cold water. They tend to be less dissolved in warm water. So global warming will definitely not help for this. Yeah, I just mentioned that oxygen is a waste product. Actually, you need to dump your electrons somewhere during photosynthesis, and during respiration. But oxygen is a waste product of photosynthesis. Essentially, photosynthesis, you can do photosynthesis with many different compounds, with sulfur, hydrogen sulfide. In those cases, you don't release oxygen. So that's not oxygenic photosynthesis. But then at one point, again, evolution made it possible to split. Instead of other compounds, it found a way of using the hydrogen in the water molecule, which is already abundant, right? This is a major innovation in evolution of life, because then this, first of all, this made possible the widespread production or widespread diversification of photosynthetic organisms. It also enabled oxygen injection to the atmosphere. So this splitting of water is not easy. We are still trying to do it, actually. Many of people are asking, can we use hydrogen sulfide in Black Sea energy source? Yes, you can, but you can already use water as an energy source. It's hydrogen in water. and life already solved that problem. We can also do that chemistry-wise. It's possible to split water, but we cannot do it as efficient as life at the moment. But I know that a lot of chemical laboratories, research laboratories, they are working on this problem. Another problem is, of course, how to store hydrogen once it's split it. In our case, in case of cyanobacteria, algae, they use things right away, so they don't need to store much. But in our case, we will need to store hydrogen, etc. But water is a big energy source as long as you manage to split the hydrogen. So oxygen is a waste product in that regard. So it's an extra in a way. And when it was introduced to the Earth system, it was probably like a very, very toxic chemical species. There's also the idea that, to confuse you further, but it's good for you to know these things. I mean, the picture we paint here was like a primitive world becoming more and more complex, right? Some people argue that actually the microbial world was a lot more complex pre-oxygen times, but then the introduction of oxygen caused large, let's say, mass extinctions in the microbial world, because oxygen is very, very toxic, actually. When you think of, if you are not used to it, many anoxic species, they cannot live under oxygen. So there's also this idea, which is very appealing, that the introduction of oxygen by cyanobacteria caused huge extinctions in the microbial world, which we don't even find in fossil record. We don't know. So we have this simple view of, yes, life evolved maybe in a hydrothermal vent or warm needle pond, wherever, and then became more and more complex. But maybe it was not like that. So with regard to oxygen, probably the introduction of oxygen caused extinction of many microbes like this. So the microbes that we have today, the anaerobic ones, maybe they are the ones left over of those extinction events. All right, let's come a little bit to the scale of oceans now. Okay, a little bit oceans and seas. When you want to think about that scale, then physics come into play. Okay, you need to now think about how water mixing occurs, ocean circulation, etc. And any actually water body on our Earth, oceans or lakes, they are called, they are stratified fluids. Stratification meaning density stratification. I mean, since gravity is acting on a vertical scale, the denser portions of water will sit beneath, but more lighter portions, low density portions will sit on top. And it happens that the more dense stratification you have, the more difficult it becomes to mix this water body. And oxygen, in this case, it's produced in the surface photic zone, and it can mix up from the atmosphere. So the surface is the source of oxygen for deep waters. If you isolate the deep waters through stratification, then it becomes more difficult to introduce oxygen. So this graph summarizes this. So oxygen, there's a lot of oxygen around, but it's not equally distributed. In a way, the resource not equally distributed. So the deep waters are always, they have this risk of losing oxygen, becoming anoxic. For life, this has huge consequences, of course. Once oxygen is absent, then of course you don't have macrofauna. You have only those organisms which can survive. Anoxia, you have all kinds of other anaerobic organisms. So that's one important aspect of hypoxia. Then once you remove oxygen from your seawater, also you have many different changes in the sediment underneath that water body. Maybe those of you studying lakes should know these things. I mean, once you remove oxygen, things come out. Phosphorus, for example, or iron sulfide even. They can briefly come out. We hear a lot about fish kills, for example, in the news, especially in Turkey, where the nature is under a lot of pressure. I believe this is mostly these sort of anoxic events. Okay, it's mostly related to some sort of toxic contaminant, but it's just sometimes lack of oxygen and this overturning of water bodies suddenly with sulfide or other reducing substances, really just suffocating fish. So it's a risk also when you consider management of the water bodies. So let's move to the ocean scale. So here you see in oceanography, we look at concentration profiles a lot, especially in biogeochemical oceanography. Here you see concentration, x-axis, and y-axis is the depth. This is also contrary to how you would plot data, right? You normally plot independent parameter like depth in x-axis, but in this case, we want to visualize it the way it really is. So here we go, even we put the 0, it's reversed. So the y-axis is the depth, and here the concentration. So these are oxygen concentration in different depths, Pacific and Atlantic. You see, different ocean basins, they are also different in their oxygen distributions, but I've just told you that the deeper it is, the more likely that you may lose your oxygen. But looking at the Pacific and the Atlantic, the deep waters, 5,000 meters, a lot of oxygen. Do you know why? Any idea? Of course, I know a couple of you have the answer, but maybe they can remain silent, but maybe others. Why do we have a lot of oxygen in currents? This is also related to Shevval, your question. How was it possible to oxygenate if it's so densely stratified? How it's possible to oxygenate the oceans? We have a lot of oxygen even in the Mariana Trench, 11,000 meters deep. Some answers are flowing. So due to volcanic activity, okay, how does volcanic activity bring oxygen to the deep? Okay, actually the answer here is again physics, as I gave you the hint a moment ago. When you scale things up in a planetary scale or even in a lake scale, you need to consider physics, mixing. Of course, in a more isolated water body like Black Sea, It's more isolated, so the circulation, water circulation is very slow in a way. But the oceans, global oceans, are quite connected by a huge set of deep water circulations, collectively called that thermohaline circulation. And in this case, the deep waters of the Pacific or Atlantic are quite well oxygenated because they form in polar regions. Polar regions being very cold, they can dissolve a lot more oxygen, and they are mixed a lot because of the cold temperatures. You don't have much of a density stratification, so what we call dense water formation happens in polar regions, and they sink and they actually bring oxygen. They ventilate all the deep waters of the global ocean. We call this global thermohaline circulation with a time scale of I think like 100,000 years or so. The whole waters of the global ocean, they are renewed actually with this circulation. So in the end, you have oxygen being physically brought to the very, very depths of the ocean. The fact that Black Sea or Marmara receives little oxygen is because they're isolated from this global thermohaline system. The Mediterranean, for example, is fully oxygenated also. Even 5,000 meters down in the Mediterranean, we have oxygen because it is slightly connected to the global system, of course, but it also its own ways of deep water forming in certain regions, and these deep water sink, brings oxygen, ventilates out of the deep areas. The volcanic activities are a very good question. It brings, it more acts the other way. It consumes oxygen because, as I'll switch to hydrothermal vents in the next lecture, hydrothermal vents bring a lot of reducing more substances, so it consumes oxygen rather than bringing in new oxygen. But let me tell you an interesting thing. There is another process, which is not volcanic process, but it's about the inside of the earth. And this is related to FS question about the microbial diversity in the deep. So you can go really deep down, even you can finish the oceans, go into the sediments, even 10 kilometers, 10 to 15 kilometers down in the sediment. Now it's crust, it's no more sediment, you have actual rocks. In the crust, there is microbial life. People are finding microbial life. Basically, a lot of questions about what is fueling this microbial life. Actually, what is fueling is, of course, part of the reason is the food that's left from the surface of the earth, but also chemosynthetic pathways. And one of the ideas is that as you go deeper and deeper, the radiation emitted from the Earth's core splits water into its hydrogen and oxygen by itself. And then, since there's some sort of splitting, then there you go. Once you have free hydrogen, that's food for especially archaea. FS question is very, very good, and that's a very frontier area. Again, another big area that you should be thinking about. How do different deep zones compare? The deep lakes, deep oceans, first of all. That's a good question. I think they are both quite diverse, but in different ways. Deep lakes, for example, probably they receive a lot more terrestrial minerals, which may enable different, for example, we talked about uranium eating bacteria. I'm sure you can find a lot more maybe diversity in deep lake sediments because of the terrestrial input. But deep ocean sediments can be also diverse. But as a general rule, I think there's a reduction in diversity, microbial diversity with depth. Primarily because you have less and less ways of supporting, metabolically supporting your communities. And as you go deeper in the sediments, in the ocean and in the crust, it's mostly archaea. Methanogens, for example, it becomes more like archaea dominated. So yeah, more archaea in the deep, truly. So that's a general tendency that people find. That's because the food sources in the very deep areas, they're either hydrogen or methane. So in that regard, and it's mostly the archaea who are dealing with, you know, those substrates in the current. So when you switch to the ocean level processes, then the lesson is physics also come into play. So that's why you always look at, we need to also learn some physics if you want to understand oxygen in the oceans. So it's interesting. Currently, this is the oxygen levels at 10-meter depth of the oceans. Don't worry about the numbers, but basically green is good, blue is good, red is even better. A lot of oxygen you see in the polar regions, right? That's temperature. So all these oxygen is being synced and injected to the deep Atlantic and deep Pacific waters, okay? So there's a lot of oxygen being injected to the deep sea from polar regions. That's why polar seas, as you know, they are warming up very fast. It's a big threat to the ocean circulation, because the prediction is that ocean circulation will slow down as the Earth will warm up, which is very, very bad news for world's oxygen. So this is oxygen levels 10 meters depth. Imagine you remove the upper 200 meters of the oceans, and now we are looking at the oxygen levels in that 200 meter. And you see now all of a sudden we have all these anoxic zones appearing. Okay, where are they? Huge areas. Eastern Pacific, you see Arabian Sea in the northern part of Indian Ocean. You see Black Sea, coasts of Africa. There are some, you know, Gulf of Alaska, etc. So actually, anoxia and low oxygen waters, they are quite widespread. It's not just because many people think it's just a peculiarity in some lake or Black Sea, and that's it. No, it's actually quite widespread. And these areas are being enlarged because of what I've just told you. And there's actual data on this. There are many type of data like this. We also have data in our institute since 1970s about oxygen levels in Black Sea, Marmara, and Mediterranean. The trend is the same. It's even worse. For Marmara, Black Sea, it's hugely worse. That's quite bad. But this is the global ocean, I think, more different data integrated. So there's a net loss. There's a very solid trend of oxygen loss. So these areas, area-wise, they are being enlarged at the moment. This is another data set from Eastern Pacific. You see, this is 1960s to early 2000s. So this low oxygen core is getting thicker and thicker and thicker, as you can see. And you see the depth at which oxygen disappears is also becomes more shallow. That's bad news for fish or other megafauna living or migrating in this zone. So the ocean life, especially the higher levels of life, evolutionary, newer species, let's say, they will be more, their habitat is shrinking in a way. So maybe that's another take-home message for you from all this discussion, is about the availability of habitats with the global change is really, really being shifted a lot. There's definitely in the oceans, there will be less and less oxic habitats for those species who are in absolute need of oxygen. Let's also briefly discuss nitrogen, and we'll do a break, switch to deep sea ecosystems. Nitrogen is also key. Let me just show this to you before. That's the nitrogen profile in the oceans, general profile. Usually you can see they are depleted in the surface, right? Phosphate, nitrate. Do you know why? Why it's zero in the surface and just increases with depth? That's contrary to oxygen in a way. Any idea? I think being ecologists, you should be really answering this. This is the same in terrestrial ecosystems too. I mean, nitrogen, phosphorus, they are primary nutrients, and they are, as soon as they are available, they are taken up by biology. And since we have more biology in the surface of the ocean, they are taken up and being used to depletion. So wherever comes in, being used, becomes part of the organisms, okay, part of the organic pool. This is the inorganic dissolved nutrients. So in biogeochemistry, we talk a lot about what is dissolved, what is particles. It can be very confusing, but that's how it is. They are different. You need to locate where your nitrogen is it dissolved, available, bioavailable for organisms, or it's not there, but maybe it's in the particle pool as a part of the organic molecules, etc. You need to be able to spot as a biogeochemist where your nitrogen, phosphorus are to be able to make inferences about the ecology. Generally, this is uptake, and due to respiration over the depths of the ocean, then this nitrogen and phosphorus becomes again available to the dissolved pool. Anyway, in the end, we end up having different profiles. Of course, the levels are different. The concentrations of nothing in the ocean is constant. It will be different depending on where you are. Okay, so don't believe in some people say because there's this much of this in the ocean. No, it will be different depending on where you are and what are the processes there. So in a very simplified form, this is the nitrogen cycle. It's quite complex, to be honest, more complex than this. But again, the presence of oxygen or the lack of oxygen is a huge player in the nitrogen cycle. Nitrogen can be, of course, as I said, in inorganic available form, or can be as nitrate or as ammonium. But they can be both be organic nitrogen as a result of assimilation or life processes. Nitrate can also be used as in respiration, okay, to dump electrons. That's called denitrification. Actually, the respiration of nitrate is called denitrification. If you hear about it, I know in ecology denitrification is a major player. When you see it, imagine that it's like oxic respiration, but just inhaling nitrate. But what is interesting with denitrification is that it occurs in steps. And one of the steps is the production of N2O, which is a greenhouse gas. So that's one caveat with the nitrogen cycle. The more anoxia we have, it's possible that the more nitrous oxide emission is possible. So that's kind of also bad news for climate. Again, nothing in the ocean is static. These are different estimates of nitrogen sources and sinks to the oceans. Now, most of the estimates show that we have more loss of nitrogen at the moment rather than introduction of nitrogen. So there's a net loss of nitrogen from our oceans. Loss, you may ask, where is it going? It's going to atmosphere, of course, primarily as nitrous oxide and nitrogen gas. And this is mainly happening in this oxygen minimum zones that I showed you, where oxic respiration is not important, but denitrification respiration is very important. So one big result of denitrification is the production of N2 and N2O. Okay, unless it's fixed back to ammonia by nitrogen fixes. That's another story. Very complex, but this is a more or less similar process set also in soils, in terrestrial ecology. So I think you need to know a little bit of these things to be able to. I had some examples from Baltic Sea, Black Sea, but I'll skip them for a moment. I propose let's do another 10-minute break, and I'd like to devote my last half an hour to deep sea ecosystems such as deep sea volcanoes that Zeral asked, and the chemosynthetic life around those volcanoes. And we will also look at some gas methane-based chemosynthetic ecosystems as well. Any question about oxygen, nitrogen, modern processes? You can also ask when you return. Okay, then if you agree, we can do another 9 minutes, let's say, so 20 past